Hi, I'm Dr. Goedele Maertens from Imperial College, and today I will be telling you how we can eliminate HTLV1. So HTLV1, or human T-cell lymphotropic virus, is a retrovirus, and one of the key stages in the replication cycle of a retrovirus is that it needs to integrate or insert a DNA copy of the viral RNA genome into the host chromatin. This reaction is catalyzed by the viral integrase, and integrase catalyzes a couple of reactions. So here schematically this is shown as a blob of integrase that recognizes in a sequence-specific manner the LTRNs of the viral DNA. And we refer to this complex as an intosome or pre-integration complex. The first reaction that it catalyzes is a 3' processing reaction where it removes a dinucleotide from the 3' ends of these LTRs. And this produces nucleophilic hydroxyl groups. Following engagement with the host DNA, these nucleophilic hydroxyl groups attack the phosphodiester backbone, linking the viral DNA with the host DNA. Now, because the integration happens across the major groove, this results in gaps upstream and downstream of the integrated provirus. These gaps are repaired by host cellular enzymes producing a stable provirus. And it is at this stage that the host and the cell is doomed, because now this cell contains all the genetic information needed to be able to produce new viruses. This cell and every daughter cell it produces. HIV-1 patients in the clinic are treated with integrase strand transfer inhibitors, or INCIs. And these drugs block the integration reaction at this step, so following 3' processing, but before engagement with the host DNA. When we measure integration activity in vitro, we do this by mixing recombinantly purified HTLV1 integrase with viral DNA mimics, double-stranded oligonucleotides that mimic the LTRNs of the viral DNA. We mix this together with supercoiled plasmid DNA, and if we have concerted integration happening, this means that 2 viral DNA ends integrate into the supercoiled plasmid DNA. This plasmid becomes linearized. If we have defective integration, only 1 viral DNA end gets integrated, we have a circular half-side product. Now, each of these products we can separate on gel. So this is an example of an agarose gel in which we have the supercoiled target DNA. This is a negative control reaction. Here we don't have any supercoiled plasmid DNA, and this is our control where we don't have any integrase present. However, if we add integrase to this reaction, we can see this linear strand transfer product appearing since it migrates at a higher side in the gel. This light band up here is a product of half-side integration, or nicked circular DNA. Now we can do these reactions in the presence of inhibitor, and this is an example where we use increasing amounts of raltegravir added to our in vitro reactions. And then we can go and measure the density of each of these bands, the products of strand transfer, and see how well these drugs are able to block, and quantify how well these drugs are able to block HTLV1 integration. Now here is an example where we plot the efficacy of raltegravir and its ability to block HIV-1 integration. So here on the x-axis we have increasing amount of raltegravir, and in the y-axis we have integrase activity. So in low concentrations of drug we have near 100% activity, and at high concentrations we don't have any activity at all. And we can determine the IC50, so this is the concentration in which we have 50% inhibition of activity, and for HIV-1 and raltegravir this is 77 nanomolar. When we do these exact same reactions for HTLV1, we find that these curves are almost overlapping. And IC50 of raltegravir is 86 nanomolar. So this FDA-approved drug that is used in the clinic to treat HIV-1 patients and to prevent transmission between serodiscordant couples and to prevent transmission from mother to child is able to block HTLV1 integration in vitro. The next step was to see whether the other integrase strand transfer inhibitors that are either in clinical trials or have been approved by the FDA to use and treat HIV-1 patients are also able to function in inhibiting HTLV1. And this is here a dose response curve. Within the y-axis here now, inhibition of integration. So at high concentrations we have 100% inhibition of HTLV1 integration. And this we show that raltegravir Elvitegravir, bictegravir, and dolutegravir, as well as these two compounds, very potently block HTLV1 integration in our in vitro reactions. The next step was to investigate whether these compounds are able to block transmission of HTLV1 in cell culture. HTLV1 transmits via a cell-to-cell mechanism using the virological synapse. So here in our assays, we used MT2, which is a persistently HTLV1 infected cell line that we co-culture with Jurkat cells. And the Jurkat cells were treated with increasing concentrations of drugs. We measured the infection by quantifying the proviral load using qPCR. You can see here that raltegravir and elvitegravir potently block HTLV1 infection, and bictegravir is even more potent. Now, to confirm that there is indeed a block in integration, we also used allo-PCR. And allo-PCR is a method where you quantify the junctions between the proviral integrated DNA and the host genome. And as you can see here, DMSO, that is our negative control. If we add raltegravir, elvitegravir, or bictegravir, we have a strong, significant drop in measuring any products of integration. And this is shown here also on the agarose gel. MT2, this is our cell line, persistently HTLV1 infected cell line that we use as a donor for HTLV1. Now, we published this in 2019 and showed that the EC50s for raltegravir or elvitegravir are between 6 and 10 nanomolars, and bictegravir is much more potent, blocking HTLV1 infection with an EC50 of 0.3 nanomolars. This is all in a similar range as how these compounds block HIV-1 infection in vitro. Now, we wanted to understand why and how these drugs are able to block the HTLV1 infection so efficiently. So in order to do this, we needed to solve the structure of integrating complex with the viral DNA. Now, to cut a long story short, we were able to do this using STLV1 integrase, so simian T-cell lymphotropic virus integrase, and colleagues from the IHARA lab were able to do this as well using HTLV1 integrase, and we come to very much the same conclusions. This is the EM model of the STLV1 intosome, and here this is the structure of the intosome using X-ray crystallography and models that we were able to dock into this EM density model. We find that there is 2 molecules of viral DNA as expected and 4 molecules of integrase. In addition, here in purple, you can see 2 molecules of B56, that is a host factor of HTLV1 integration, which we really needed to be able to form stable synaptic complexes. So, as an overview of the STLV1 intosome, we have 2 molecules of viral DNA, 4 molecules of integrase, and 2 molecules of the host factor B56. We took this a step further and solved the structures of the intosome in complex with raltegravir and bictegravir. These 2 integration and transfer inhibitors that are FDA approved and that function very well in blocking HTLV1 infection in our tissue culture model. And what we observed is not that surprising, but although the sequence conservation within the active site between different retroviral integrases is very low, the binding mode of these drugs is highly conserved, explaining why these drugs are so potent in blocking HTLV1. A couple of years ago, cabotegravir, which is a long-acting integrase strand transfer inhibitor, was approved by the FDA to use in prophylaxis with HIV-1 patients, and we show here that cabotegravir as well potently blocks HTLV1 integration and transmission in vitro. This is an allo-PCR showing that in the presence of cabotegravir, we block HTLV1 infection. We went back to dolutegravir. This is one of the earlier integration and transfer inhibitors, second generation, that we showed previously were able to block HTLV1 integration in vitro, and we went back to check whether this compound is able to block infection in cells. And dolutegravir is currently one of the first-line inhibitors that are used to treat HIV-1 patients. And we can show here, using these dose-response curves, that dolutegravir potently blocks HTLV1 infection with an EC50 of 0.25 nanomolar. And this is confirmed here by allo-PCR. It's not that surprising that dolutegravir is as potent as bictegravir in blocking HTLV1 infection because their structures are highly similar. Now, TAF, or tenofovir alafenamide, is an RT inhibitor. So RT is one of the other viral enzymes that are critical to establish infection. It's the enzyme that converts the RNA genome into double-stranded DNA. And TAF is a product form of tenofovir. As we show here, very potently also blocks HTLV1 infection. The EC50 is higher than bictegravir, dolutegravir, but it's still very much a very potent inhibitor of HTLV1 infection. If you look at the structures, structural comparison between HIV-1-RT and HTLV-1-RT, so here for HIV-1 we have the structure available. For HTLV-1, this is an alpha-fold structure prediction. And although you can see there are some similarities and differences between the structures, if we look at the active sites, here shown in orange, we can see that the residues that are critical for the interaction with tenofovir are 100% conserved between HIV-1 and HTLV-1, explaining why this drug also potently blocks HTLV-1. So in a table, we have 6 integrase transfer inhibitors, long and short acting, that are very potently blocking HTLV1 infection. And we have 2 NRTIs, TAF, which is a first-line inhibitor being used in the clinic that blocks HTLV1 infection. So it is time to do some clinical studies. We can do some retrospective studies. Do we know if treatment with HIV-1 PrEP prevents transmission of HTLV1 in HIV-1 and HTLV1 co-infected people? Let's do some prospective studies. Do integrase transfer inhibitors and TAF prevent transmission between serodiscordant couples and people who inject drugs? Do dolutegravir and TAF block HTLV1 mother-to-child transmission? We know that both of these drugs are safe to use in pregnant women. And can we use cabotegravir or a combination of dolutegravir and TAF in neonates? HIV-1 clinical trials are often done in places where HTLV1 is endemic. It's important that HTLV1 incidence is included in these studies. Why continue to ignore HTLV1? HTLV1 causes disabling and often fatal diseases. If we can prevent transmission, we should do it. And to finish off, I would like to thank the people who are involved in this work. Mpatso, Bethany, Michal, Jordan and Teresa all contributed to the work on INSTI and the RT inhibitors in vitro and in tissue culture. The CRI-EM work was done in collaboration with Peter Cherepanov of the Francis Crick Institute, and the data collection was done at the LONSEM. And of course, we're thankful to our funders.